Good morning and good afternoon, everybody. Thank you for joining today. We're very excited to have our panel-based tissue companion diagnostic development and image analysis in a regulated space discussion with everyone today. You have myself, Meredith James. In addition, we have Susanna Corey-Torrey, regulatory consultant, Roberto Giannani, the Chief Medical Officer with Flagship, Mike Kagi, Senior Director of Translational Medicine at Leap Therapeutics, as well as David Sidransky, Professor at Johns Hopkins School of Medicine. So during today's webinar, we'll be discussing companion diagnostic development with an emphasis on oncology in particular, what those regulatory approval pathways entail, as well as points to consider for any tissue-based companion diagnostics. Later, on top of that, we'll be discussing image analysis, performance criteria, and additional regulatory considerations that you might want to take into consideration, particularly as you move forward into trying to get some of these things accepted by the agency. So companion diagnostics are used to support the safe and effective use of its corresponding pharmaceutical product. Susanna, did you want to jump in and talk a little bit about what this looks like?
Of course, thank you, Meredith, and hello, everyone. It's a great pleasure to be with you today. Companion diagnostic is an in vitro diagnostic test device by regulations that provides information that is essential for safe and effective use of a therapeutic product, helps to identify patients that are suitable, eligible for particular targeted treatment, and also support monitoring of the therapeutic response. They're stipulated in instructions for use for both companion diagnostic and corresponding therapeutic products, the key attributes being safe and effective use. Next slide, Meredith. Ideally, therapeutic product and corresponding companion diagnostic are co-developed, and they may start at the same time, but the speed as the drug development, for example, proceeds may differ later on compared to that of companion diagnostic. Often contemporaneous regulatory approval of both may not be possible, and that is recognized by FDA. Common situation is that the approval of therapeutic product usually occurs first, and then approval of companion diagnostic subsequently is taking place, requiring the potentially revision of the label of therapeutic product in the post-approval process. New marketing indication is possible for approved drug with new diagnostic, also in post-marketing period. Regulatory oversight in any case is risk-based.
Thanks, Susanna. Before we move on to the next slide, Mike, I'd like to get your thoughts from an industry perspective. When does it make sense to start investing and really looking into this companion diagnostic pathway?
Yeah, I can. Yeah, thank you, Meredith. And I'd just like to start by saying it's definitely a pleasure to be with everybody today. Yeah, so I can also touch upon that a little bit more on the next slide. But, you know, at a very high level, I mean, my recommendation would be to start as early as possible. You know, it is, you know, quite a lengthy process going from a hypothesis all the way through CDX development. And, you know, the earlier you are beginning to think about this process, you know, you will have more time to react to inevitable issues that come up, and, you know, really, you know, kind of be able to plan for the future. And, you know, I think this, you know, this slide right here, I think it really, you know, kind of highlights, you know, kind of the development path for both a companion diagnostic and drug, you know, and drug development. So, and like I said, you know, first, you know, kind of a couple key points I want to make here. One is, you know, like I said earlier, definitely start early. And the other one is that, you know, your biomarker companion diagnostic development really should be intimately linked to your, you know, to your drug development. For instance, this kind of makes sense. If you are, if you're developing a drug where you are planning on using a biomarker strategy, where eventually you're going to be stratifying patients or using that biomarker to actually select or identify patients, that assay is going to have to be, eventually it's going to have to be FDA approved. And you're going to need that approval at the same time that your, drug, your small molecule or biologic or whatever you are developing is also approved. So you really have to kind of work hand in hand here. And what I'd like to do is kind of give a high-level overview of the process that we've taken here at Leap Therapeutics for the companion diagnostic that we are currently developing for one of our lead oncology assets. And that is a therapeutic antibody called DKN-01 that targets a secreted protein, DKK1. So, what we, basically did, I should say you really can kind of divide this kind of CDX development up into multiple different stages. And the first stage is, is much more the kind of non-clinical, hypothesis generating stage where, we sat down and basically, came up with some models and some hypotheses on what potential biomarkers could be for our drug. In our case, we had a variety, and one of the most obvious is the target of our drug, this protein DKK1. We knew that this protein DKK1 could really drive tumor growth. So for us, it wasn't too much of a kind of leap, no pun intended, that being the name of our company, to really hypothesize that patients that are producing more of this protein in their tumors are the ones that are going to be able to respond. So once established this hypothesis, the next step was to, determine what assay we were going to use to measure DKK1 levels. And in our case, we tried, IHC for protein, couldn't get that to work, and then moved on to this, RNAscope assay to measure DKK1 mRNA levels. And what I say at this point, you want an assay that's going to be reliable, but it doesn't have to be validated, verified extensively. Like I said, you do want to believe in it, but to really take it to that next level, which I'll touch upon later, such as a laboratory developed test, that's much more a financial commitment. And for us, we were testing a variety of different hypotheses. So, we wanted to make sure we had what we thought was a solid assay, but didn't have to be done to the kind of the rigor that you need later on during your clinical development. Really think of it as a kind of a research use only assay. So, we did some internal validation, got this assay up and running, and our next step was actually moving into testing some clinical samples. And this was basically going to be a retrospective analysis from tumor samples that we'd already collected during a phase 1 trial. So once again, really trying to kind of parallel our clinical development of our drug. So, we took this RNAscope assay, ran it on these clinical samples, and you were able to show that patients that were expressing high levels of the target of our drug, those were the ones that were driving the most clinical benefit. And after seeing this data, we were at another inflection point. We saw this data, we thought it was strong enough to actually, kind of move this assay kind of along the next step of the companion diagnostic pipeline. And for us, that was to actually get the assay to a level where we could use it to prospectively screen for patients. So this is, going to be, kind of a fairly substantial jump in the assay. For us, it required a partner such as who we started working with, was Flagship Biosciences. And we really needed to take our research assay and move it, from that kind of research grade to what we are now calling a laboratory developed test. And this is, really, an assay. This will then be an assay that is much more validated, demonstrated to be reproducible, accurate, sensitive, and specific, all validated to CLIA/CAP guidelines. Basically, you want the assay at a place where it can be run in one single lab, and it can be used to prospectively identify, patients for your clinical trial. So, working, and and I do want to point out, this is something you just can't do overnight. Developing an assay, getting it to a laboratory developed test level does take some time. And another thing to keep in mind, going back to like really paralleling your drug development, this is important. You also want to start this early because, in our situation, this assay, it needed to be up and running for us to actually enroll patients. And, especially for a smaller company, that was very important for us to have it running so it didn't actually delay the clinical development program. So, we got the assay up and running, and now we are currently prospectively screening patients for a, phase 2 trial that we are running right now. At the same time, you still have to, even though this trial is ongoing for us, we are, still planning for success, and we are starting to think more about the next step as we move further along the CDX development. And that next step is really taking our laboratory developed test and really transitioning it into a registrational in vitro diagnostic. And this is, you can think of this as an assay that is, that it will be locked down. Hopefully it'll be the assay that the test and assay that you use after approval of your drug. In order for this to happen, there needs to be additional verification and validation of the assay that is done. You move to a situation where you're not really research use only, you're investigational use only, you're using GMP reagents, and the assay, in our case, and probably the case for most people, the assay has to be shown that it can be run in multiple labs to support like a global registrational trial. And I should say this is another big jump, the LDT to the CDX. It's you're investing a lot more money in the assay, so you really want to be sure that you're ready to do it. But at the same time, it takes time to kind of move from that research use only assay to investigational use only assay that you can use in the registrational phase 3 trial. So you do have to be planning ahead, and it does come down to how much money you want to spend up front, and how aggressive you want to be on it. So really, I know really in summary, I think my 2 main points are start early and really parallel your clinical development as you move along the companion diagnostic pipeline.
Well, thank you so much, Mike. I don't know, David, if you want to jump in here and talk a little bit about how we design and incorporate sample strategy support from an oncologist perspective.
Sure, thanks, Meredith, and it's a pleasure to be on the webinar with everybody else. So the important thing really is to make sure that it works in terms of your needs. In general, it's important to try to start at the same time to think about what your CDX is going to look like as you're developing your therapeutic. It helps you think about what kind of samples you're going to need, and to be able to access them as early as possible. Whether it includes blood samples, for example, that you have to take that are not available, or fluid samples, or special types of tissue samples that have to be taken. The more you understand what your needs are and when you need them, the more important it's going to be to try to strategize early on and try to work together as you think about your therapeutic and diagnostic strategy. The nice thing about tissue-based diagnostics is that, as a matter of proof, almost every single patient has had a biopsy and has tissue available, often in paraffin, obviously, to be able to make the diagnosis, so that you don't have to maneuver a lot to get an additional kind of sample. And I think it's one of the great advantages of tissue-based diagnostics. From a development point of view, if you can't think about it at the same time and get those samples prospectively, that's okay. It rarely happens that everything works out. So sometimes you have to think about your diagnostic after you've developed the therapeutic, and you just have to think through it together with the FDA of how that's going to work. You can sometimes go retrospectively back into samples. Sometimes you have to have an additional trial where you collect the samples and then do the tissue-based diagnostics. But again, those are all in discussions with the regulatory agency. In the end, I think the most important thing to the take-home lesson for me is that everybody, I think, should hear is that you really want to do it as early as possible. As just what you heard, that the minute you think that the tissue-based diagnosis is going to help you understand how a patient is going to develop a therapeutic, how they're going to take it, when they're going to take it, and when it's relevant, the more relevant it becomes in terms of the development of the diagnostic, and the more important it is to think about it then. If you think about it way after the fact, it can hurt you in the sense that it may be very hard to access the samples, and it may be hard to go back in a reasonable period of time with a lot less money than if you think about it at the beginning. So I think the message is to try to think about it as early as possible, to talk to the regulatory agency about how that trial is going to look, not so much from the therapeutic point of view, which has already been discussed, but more from how that diagnostic is going to be developed so you can plan the right strategy.
Oh, great point, David. Thank you. So, Susanna, would you feel like jumping in and talking about regulatory considerations specifically around oncology companion diagnostics?
Of course, and to David's point, it's really important to engage in an early dialogue with the regulatory agency from a strategic level, and then expand into implementation of that strategy. In oncology, that is particularly important because of the large number of targeted precision medicine-based drugs, and the risk level associated with the companion diagnostics with oncology drugs, because oncology drugs have inherent risk profile. They are imparting critical life benefit to the patients, and the process of collection of the tissues necessary for laboratory developed test and later on companion diagnostic are medically invasive procedures like biopsies. Since the regulations of the companion diagnostics is risk-based, automatically this puts oncology tissue-based diagnostics, companion diagnostics, into so-called category 3 of risk, which is considered high risk. And common scenario is that a pre-market application is required and a process of clinical trials to demonstrate required performance as related to the safe and effective use of therapeutic product. Follow-on approaches, further refinement of an approved companion diagnostic can rely on a less stringent regulatory approval pathway, such as 510(k) clearance, where the originally approved CDx serves as a predicate or a reference IVD test.
Thank you, Susanna. Mike, did you have anything to add here?
No, I think actually Susanna covered it very well.
Great. All right. So talking about bridging your lab-developed test to a clinical trial assay, Mike and Susanna, I think this is a good place for you guys to jump in as well.
Yeah, I can definitely comment on this and talk a little bit more about our own experience too. So so I said earlier, we've currently developed a laboratory developed test, and that is actually being run by Flagship Biosciences currently to prospectively screen patients that have high tumoral expression of the target of our drug for our phase 2 trial. Of course, at this time we are, like I said, we are planning for success. We believe this trial is going to be successful, and in that case, we're starting to think about how we transition this assay from a laboratory developed test to a registrational clinical trial assay that can be run for a basic registrational phase 3 trial. And as I mentioned earlier, there are some big jumps here. I mean, one is we have to move away from research only reagents to GMP reagents, and this can take time. For us, since we are using an RNAscope assay, this does involve working some with ACD, the developers of the technology, to actually manufacture multiple GMP lots of the reagents that will be used. The other big jump is we are going to eventually want to run our test in multiple different labs. We're going to have a global registrational phase 3 trial, and we are planning on having at least 2 different labs. And this really kind of moves it away from the kind of the laboratory developed tests into more of a investigational use only, where you have to demonstrate that the assay is not just reproducible, accurate, sensitive, and specific in one lab, but in multiple labs. And that is definitely a higher bar to go over. The other point I want to make with this assay, with the registrational CTA that we want to run for our pivotal phase 3 trial, we want that assay test to be at a place where it is as locked down as possible. We are hoping that we can start our trial, with an assay/test that we will not have to modify earlier. And this is being done to really kind of decrease any potential risks that we could have with the FDA, or really any mitigate concerns where if we had to modify the test halfway through of our phase 3 trial. I mean, that is something that we definitely want to avoid. I also one of the final point I want to make, there can be changes in your test going from the LTD into the CTA right before you start the pivotal phase 3 trial. And in order to demonstrate comparability, we are saving as many clinical specimens and slides as possible from our current phase 2 trial, so that we, if we do have to tweak our assay somewhat, then we can go back and actually run the modified assay on the slides and demonstrate that the modified assay is performing at a comparable level as the one that we originally ran. So one final point too, when you do go into a registrational CTA, this also does require another level of verification and validation. It is more rigorous than what you have to do for just a laboratory developed test. And this will require running your biomarker assay on multiple different samples. And these can be samples that you can commercially acquire. What I would like to say though is that a lot of times it can be challenging to find these commercial samples. And that is something you want to start thinking about earlier. Ideally, you want to acquire samples that are going to be very similar, or if not identical to the samples you'd be testing for your phase 3 trial. So for us, it's buying FFP resections of the oncology indications that we're interested in. A lot of our patients will have endoscopic biopsies, so can we acquire enough of those? Some patients might have core needle biopsies. Can we find those? And I can say from my own personal experience that this can be challenging to do. It can take a while, and not all of the tissue samples that you might want could be available. In which case, that might necessitate a kind of discussion, with the FDA to either figure out how to best address that, and what the risk mitigation can be going forward, and what the FDA is comfortable with there. So really in conclusion, I'll say I just want to kind of final point, the jump from an LTD to a kind of registrational CTA, it is a big one. It's a substantial financial investment, and definitely takes a lot of time. Also you have to acquire a lot of different tumor samples, so it is something to not be taken lightly. And I said it earlier, and I don't want to be beating a dead horse there, but start thinking about it early, so that you can be as prepared as possible.
And I'd like to add to David's excellent summary on the challenges, that in addition to it, rigor and level of quality of during the development of the test, let's say laboratory developed test, it is associated with certain quality and standards of performance, which is certified under clear conditions in order to be eligible, the test to be used for prospective enrollment of patients that have expressed that particular target that the drug is is impacting on. So that also requires understanding on side of the sponsor, what does it take to develop and validate laboratory developed test under CLIA standards. And this is something that is definitely part of the package that your IRB for the trial will be looking at, and will have as an insurance that the test, despite the fact of the laboratory nature of development, still satisfy high level of quality standards that will later on further be developed and expanded.
No, thanks for that, Susanna. And just a couple notes here. There's a new framework in the EU for lab-developed tests, and we don't necessarily have to go into some of those changes here, but if you have questions about it, feel free to drop it into the chat, and we can talk a little bit more about that at the end.
Yes.
All right. Susanna or Mike, did you want to jump in here and talk about the approval pathways for new IVDs and companion diagnostic assays?
Mike, you want me to take the first step and then you can expand?
Yeah, I'll let you take this one, Susanna. This is much more in your wheelhouse than mine, so.
Okay. I think I already mentioned that the classification of the companion diagnostics and regulatory oversight is risk-based. So the regulatory approval options are also risk-based and depends on is there already an approved test on the market that can serve as a reference test, or this is a new drug, a new companion diagnostic, which will have to follow different pathway. In general, there are 3 different regulatory pathways for a companion diagnostic and IVD. I mentioned 510K pathway, which practically uses existing reference product on the market as an equivalent or so-called predicate. In which case the focus of the regulatory submission is really to establish the equivalency or connection with the predicate. However, no clinical trials are usually required under this regulatory option. Most of the new IVDs and companion diagnostic will follow pre-market approval pathway, which practically will require a clinical program that will be incorporated into drug program. And that's a complex process depending on the target, depending on the test, and depending on the disease, and it's most expensive and time consuming. And there is another pathway that is called de novo reclassification, where there is no predicate IVD that exists on the market. However, the new IVD or CDX is of low risk or moderate risk, in which case, again, no comprehensive clinical evaluations will be required. Most companion diagnostics of new drugs will follow the PMA pathway, and for that one, early consultations with FDA have to be scheduled and discuss the strategy. Sample retention, Mike already pointed out. So please have that in mind. Okay.
Thanks, Susanna. Roberto, did you want to talk a little bit about the clinical validation process here?
Yeah, sure. Good morning or good afternoon, everybody. This is a pleasure for me, and thank you for joining us in this discussion. So the clinical validation process of this test is, of course, very important, because you want to make sure that everything you do is consistent with the level of quality necessary for this very complicated and serious undertaking. So from the point of view of the clinical validation of the analytical and clinical validation of this test, one thing that is extremely important is actually to have a very large amount of number of specimens. And this is actually true for two reasons. The first reason has to do with the analytical part of the validation, right? So even in the situation where one is actually dealing with just one tumor indication, the heterogeneity within each indication can be so large that only utilizing a very large amount of specimen will actually yield a different type of morphology that will create a solution that can be actually useful in the real world. And that's the analytical reason. The second reason is that, ultimately, even after you have completed the analytical validation of the test, so in other words, you have made sure that the test perform as you intended to perform, there is the so-called clinical validation of the test, right? So to basically make sure that the test correlates well with predicted outcome, or is able in general to predict outcome, because at the end of the day, that's actually the real testament of the usefulness of the test. And to be able to predict clinical outcome and to verify clinical outcome, you need a very large amount of specimens indeed.
Thanks for that, Roberto.
Yeah.
So within the context of companion diagnostic development, image analysis incorporation is still very open-ended, but options can be validated for clinical prospective use. Image analysis can decrease time and overall cost due to appropriate patient selection in these large data sets, which we've discussed previously, and it's rapidly becoming a critical tool to support pathologists due to the increasing complexity of the unique biomarkers that we continue to see, particularly in immuno-oncology. As the development moves into later phases and more sites open up, you ensure much more consistency across reads and ensure that you're getting the best data possible. So that being said, I think it would be good to talk about the IA-based companion diagnostic approach versus traditional pathology. So I don't know, Roberto, if you want to start a little bit with your experience.
Sure. So, of course, I am myself a pathologist, and I'm going to have only good things to say about traditional pathology. Traditional pathology is a time-honored approach dating back from the 16th century when the microscope was invented, and it's been extremely useful. But one thing to keep in mind that traditional pathology is mostly geared, and has been geared for all this time, to actually reach a diagnostic consensus between pathologists over what the tumor actually is. And that's actually very different than what pathology is actually now called to do in the era of target oncology therapies that can be very complicated and can require a lot of information for predicting the usefulness. Traditional pathologist doesn't really do as good a job as it did in the diagnostic work. And hence, in this era of personalized medicine, the digital image analysis come true. In truth, though, we have to keep in mind that these computer-based digital image analysis are still rooted very significantly in the traditional pathologies in terms they try to recapitulate the approach of traditional pathology, and also the pathology is involved at every step of the way. But there are things that the computer can do better than pathologists, like being consistent, so increasing the precision of the outcome, and quite frankly, being more accurate when it comes to actually count cells or deal with a very large amount of data. So I think in the last analysis, the cooperation of the human, the pathologist, and the digital domain will actually yield the most appropriate way to negotiate all the information that are needed for these trials.
Yeah, no, absolutely. I agree, Roberto. David, did you want to talk a little bit about, from an oncologist perspective, what makes sense here?
Sure. Yeah, and I think I really want to build a little bit on what Roberto said. Look, I think in the end, for an oncologist, we understand that an IA-based CDX is going to be designed to either be equivalent or perhaps a bit better or substantially better to, normal pathological analysis or approach to tissue. However, I think it's pretty clear then when you're going to send a sample across the country, potentially, processed in a different way, arriving at a different laboratory, seen by a different person, having to be done manually, there's going to be a lot of issues regarding reproducibility and what the answer really means. And I think for an oncologist, the computer algorithm really is meant to augment or improve the ability to quantify the biomarker from a microscope, and in a manner that is consistent with a pathologist trained approach, but obviously has better precision and reproducibility over essentially, different pathologists trying to come up with the same answer. In some cases, I think it may be the only adequate level of performance required to stratify patients. And I think that way really differs substantially from just manual review of a slide. I think the other thing to think about, just looking into the future, is that the complexity of targeted oncology therapies, and we're looking at new approaches like antibody drug conjugates or ADCs, immuno-oncology modulators. I think they're going to drive more and more reliance on image analysis due to the complicated biology, the mechanism of action, and really the complexities of the way tissues or other fluid samples need to be assessed. So I think that IA-based CDX is a way to really improve substantially over the way we currently look at slides. And from an oncologist point of view, when we're thinking about staining a slide and deciding whether patients will get a life-saving therapy or not, it is critical that that answer be as accurate and as precise as possible. So that's the way I see IA, and I think that as we continue to develop IA, I think it will eventually replace much of the manual assessment that we do today.
Yeah, and just a comment, like I 100% agree with what David said. I think IA is going to be the way of the future, and we just see with a lot of the even some more of the retrospective analysis that are coming out, people are doing more and more multiplex IF, multiplex assays, and it's just getting to a point where pathologist isn't going to be able to manually score these. So when you're dealing with such a level of complexity, the only way I think you will be able to reproducibly and accurately analyze this data is through IA approaches.
Yeah, so I agree, Mike, and I think obviously what you guys are working on now at LEAP is a great example of that. So I have your case study pulled up if you just want to walk through, give us a high overview of maybe what some of the challenges were and how image analysis was able to solve that problem.
Yeah, no, I'd be more than happy to, Meredith. And in our case here too, we were, in theory, our assay is pretty straightforward. Like I mentioned earlier, it's an RNA-scope assay, technology developed by Advanced Cell Diagnostics, where you're basically measuring mRNA levels. The nice thing about this technology is, it's exquisitely sensitive. In theory, it can pick up 1 mRNA per sample, which can be hard to find something necessarily that sensitive for IHC. It was an issue that that we ran into. So basically the one problem with, or potential problem with RNA-scope is that it can be very challenging for a pathologist to manually score. And really, how you score this score RNA-scope data is that you have to really count the number of dots in a cell. So the more dots there are, the more expression that you have, and you have to really be able to identify these cells that have dots. And what that requires is the pathologist to really zoom in onto the tissue at a high magnification to identify a cell that has dots. Then the pathologist, kind of has to zoom out, move to another section of the tissue, and continue to do that. With all this zooming in and out, I mean, it's very easy for the pathologist to, kind of lose context of the whole tissue. And and it's kind of different than IHC, which a lot of times you can just score. The pathologist can look at a slide and just score at a lower magnification. So because of this, we were, kind of concerned about reproducibility. And what we wanted to do was try to develop a, kind of an image analysis approach that could basically quantify this RNA scope signal. Like I said, RNA scope is in some ways kind of perfect for this because it forms these nice kind of foci in the cell. So basically, all you really need to do is develop an algorithm or software that can identify tumor cells and then count the number of foci in the cell. So, what we did some initial work to, looking at this, and we were able to show that that the image analysis was actually much more reproducible than just having pathologists score slides. And, in a lot of ways that makes sense. If you have an image analysis algorithm that is, performing well, it should perform the same every time and should be reproducible. So by kind of developing this image analysis, I think we, really were able to, kind of alleviate this, this kind of reproducibility, concern that, that we had. I do want to mention, as part of the CLIA CAP validation, you do have to show that you almost have to do it in two steps. You do have to show that the wet assay is performing well. And then subsequently, after that, then you also have to show that the image analysis is, is working well too. But, like I said, I think image analysis, at least for us, it's been, much more reproducible. And, as I mentioned earlier, I think you're going to see it being used significantly more, more in the future, especially, as you see more of these complex biomarkers coming down the pipeline.
Thanks, Mike. Roberto or David, do you have anything to add here? Okay.
I think Mike just summarized this very well.
All right, so moving on to image analysis in the context of a medical device. Roberto, I think it would be good for you to kind of talk through some of the, the new guidelines that are being discussed and what does this mean from a regulatory perspective?
Right. So essentially, in terms of regulatory perspective, one is to keep in mind that once you transfer a wet assay to a digital assay, a digital solution, you essentially create a new assay. So all the traditional steps that pathologists are very familiar with, they go into the validation and the quality control and the regulatory aspects of the traditional assay are still true with the notable change that now the degree of complexity has actually grown. And this is even more true when you try now to make this assay in a device. So you're trying to go from an LDT to a device because the degree of complexity and the degree of work that has to be done in order to validate the assay and in order to keep doing the QC of the assay while the assay is working is very, very high. So the classification sometimes of the assay and the type of risk associated with each of these devices is also complicated because we are a little bit in a new territory here, right? So there is no very well established precedent to classify things in one way versus the other in terms of the risk. So the things to actually essentially keep in mind is that, one has to use the guiding principle of QC when dealing with this assay, and has to have a very rigorous process of validation of the assay that actually, and there is a little bit of a paradox, keeps the pathologist interpretation as the gold standard, but also is very well aware that in some cases this assay may be dealing with pathologist impossible endpoint, and therefore by definition one cannot use the pathology as the guiding light. And here, I'm touching a little bit of what David was mentioning. Now one would have to resort to maybe clinical outcome measure as a true validation of this assay. So the regulatory pathway involved into this kind of decision may be indeed very complex, even after one has dealt with the complexity necessary to analytically validate this assay. And I would be happy to take questions if we want to go in more detail, so what it actually takes to for the analytical validation of this digital assay.
No, thanks, Roberto. I think, another key point here is, a lot of times within the context of clinical trials, you're getting very limited tissue due to the impact to the patient. But again, that's where image analysis can really help to support and drive some of these large data sets by really squeezing as much data as possible out of these very small sample sets, and being able to integrate pathology support into that IE tool is critical here. David, I'd be curious to hear your kind of thoughts on the clinician impact around this. Oh, did we lose David? Apologies. So we'll move on and talk about the current regulatory approach for image analysis-based companion diagnostic approval. Roberto, again, did you want to jump in here, given this is something that you do every day?
Yeah. So, and again, to conclude a little bit what we were talking about before, it's in terms of now creating the regulatory and analytical framework for this assay. One actually has to keep in mind that there is a lot that goes into each phase. So, starting from receiving the images and make sure that the images have been scanned with a digital scan, with a scanner that is actually compatible with the digital solution one has created. Then going on to the development of the algorithm and the verification of the algorithm vis-a-vis the manual pathologist. There is a lot that one essentially has to do. And what are again the regulations that deal with the image analysis? Well, essentially, as I mentioned earlier, they are not significantly different from the regulation that apply to every assay, right? One is to make sure that there is the analytical sensitivity and the analytical specificity of the digital image analysis are indeed acceptable. And one is to define what acceptable is in the context of the intended use of the assay. And essentially, ultimately, one will have to say if the digital assay provide enough accuracy and enough precision to be consistent with a clinical use of this assay. There are, of course, several predictive technical biomarker approved by the FDA and the CDX, and this is only though in terms of the wet assay, and you can see them on the slide. But of these, only one, the human epidermal growth factor receptor 2, R2/νu, in the context of breast and gastric cancer, has actually an IA quantification. And I think this very fact illustrates that this is a new field, and that until now this has been considered a fairly complex undertaking. While it's true that this is a complex thing to do, the thing to keep in mind that generating a digital solution for this marker not only provide you with a better accuracy, with better precision, for the reason that we had discussed earlier, and potentially also, by the way, with a better ease logistically just performing the assay. But it's bound to also give you an additional large amount of data that simply could not be obtained by the traditional wet immunohistochemistry. For instance, if we are talking as an example about PD-L1, a traditional assay may give you some information on the distribution of the PD-L1 in addition to the quantification. But the digital assay can tell you exactly how many lymphocytes are positive for the marker, how many macrophages are positive at the marker, what level of positivity is in the macrophages in both the tumor and the stromal compartment, the TME compartment. It can tell you where are the positive lymphocytes in relation to the tumor nest, where are the macrophages in relation to the tumor nest, and so on. I could actually go on forever. So in summary, these digital assays are not only logistically more useful, but they can also be a lot more informative. As I said, you know, the regulatory framework for the interpretation of these assays is in continuous evolution, and Suzanne can speak more appropriately than me about this. But I think, you know, the extra effort is well worth when one considers the amount of information that this assay can give you.
Yeah, thanks, Roberto. I don't know if Suzanne, if you want to jump in a little bit and talk about some of the kind of regulatory gray areas and challenges in trying to embed your image analysis solution into regulatory approval, given there's no current precedent.
Right, it is indeed a gray area, but it's also a rapidly expanding area because of the rapid drug development and you know, bringing to the market a larger number of targeted medicines in oncology and in any other therapeutic area. So far, we do not have solely image analysis cleared as a companion diagnostic by FDA, but there are tests, there are in vitro diagnostic tests with image analysis quantification algorithms embedded. They're cleared by FDA, and the slide lists some of them. So it's predicted that this process will just expand, and it's all based on the rigor of the quality standards of testing and reproducibility and reliability, starting from analytical validation, going through and ending up with the clinical validation and demonstrating the performance of companion diagnostic. It also very important what is the contribution of image analysis to informing clinician for making some critical medical decisions, and it's all about the risk. Okay, so that's the background for either the traditional companion diagnostic or image analysis based companion diagnostic, and whether this can be verified by a pathologist or healthcare provider or not. So that will be, I think it's these gray areas will be worked out by regulatory agencies. FDA is working actively. A guidance that they issued a year or so about the tools for making clinical decisions is very indicative that they would like to work with the sponsors and industry and academia how to approach the best regulatory oversight, especially for image analysis. Is it maybe even situations like image analysis software, maybe only certain functions may be fitting right now in the traditional criteria for regulatory oversight as a device versus the others which are not. So these are the things that regulatory agency right now putting efforts to clarify. And Flagship is part of that process as well.
Thank you, Susanna. All right. So we only have a few minutes left. I wanted to run through and see if anyone has any questions. The slides will be made available within 24 hours, as well as a recording. If anyone is interested in a consultation with any of our experts that were on today, we will have that available as well. Feel free to put that in your response. And let me pull up some of the questions right now. So as I mentioned, we will be sending out a copy of the slides. The first question that I have is, can you provide an overview of image analysis at a high level? I think, Roberto, that's a good one for you to address.
Okay. So essentially what image analysis does is replace some of the things, but not all, that are traditionally done by a pathologist. The first step in image analysis is to actually create something that the computer can now analyze. So essentially you have a generation of a digital image that the scanner creates. By the way, one other thing that is happening is that the pathologists more and more now are relying on digital image analysis just for their own interpretation, right? So the creation of digital image analysis is also germane to manual pathology, because now the pathologist can use it as a substitute as a glass slide. But the next level that the image analysis does is to essentially analyze the data that are contained in each slide. And the first step is to do what is called the cellular recognition. So you have to create an algorithm that basically identifies which objects in the slide are cells and which objects are not cells. And once you have done that, you're able to quantify the cells. And the second thing that you have to do, you have to essentially define compartments. For instance, the tumor compartment, defined by the aggregates of the neoplastic cells and every other cell, such as lymphocytes or macrophages, that may be contained in the compartment. And the stromal compartment, the essentially support tissue that is around which the tumor is organized. Once you've done that, you can now quantify different types of cells, lymphocytes, macrophages, tumor cells in each compartment. The second thing that usually is done by image analysis is to quantify the intensity of the staining. So the staining that has been performed by wet chemistry can now be quantified by the computer, and essentially is quantified in two main ways. There is an enumeration way. So in the case you simply want to know how many cells are positive for a given marker, you're not interested in the intensity of the marker. And there is the quantification way, in which you want to know the intensity of the marker and relate that to the protein expression in cases of traditional immunohistochemistry, or RNA expression in cases of in situ hybridization. Essentially, that's what the image analysis does. Everything else is actually derivative endpoint, in which you can rearrange this basic concept and create some more sophisticated endpoint. But that's a high-level view of IA, I think.
Yeah, and that was from Lauren at CGen. If you have any additional questions, feel free to reach out and we can set up a call for you to talk with Roberto directly. I think we have time for one more question. And Mike, this might be a good one for you and or Susanna. Question is, what's the difference in planning between phase 1 and phase 3 from a trial perspective?
Do you want, I guess I can start quickly, Susanna. Yeah, I'd say just, phase 1 trial, it's, like I said earlier, it's much more exploratory. It can be retrospective. Assay doesn't need to be nearly as well validated. You want a good, biomarker assay that you're going to believe in. But, I wouldn't go crazy in, having a really well rigorous validated assay, because that costs a lot of money. And usually early on in phase 1, you're testing a lot of different hypotheses and could be running multiple different assays. So you're just not going to have the resources to do all of it. Phase 3, your assay and test is pretty much locked down. So that's the one you're banking on. So at that point, much more rigorously validated, verification has been done, all GMP reagents. You're out of research. You're into investigational use only, and hopefully well on the path for a CDX. So yes, that's how kind of I would split it. Much early and less expensive to later and more expensive and much more validated.
And I can add to Mike's commentary that practically once you get into phase 1 clinical trial, that implies that you need to have your biomarker strategy testing already in place and hopefully discussed with FDA in detail, so you have clarity how you start collecting the tissue samples, how you store them, and everything that is related to it. Now, also depends on the attributes of the investigational drug, potential anticipated risk-benefit profile or anticipated risk, especially. That may eventually push you toward a little bit more advancing into the assay qualifications and validations. And there are now phase 1 trials that use practically this IVD, for example, for a prospective enrollment of patients. So you have to have the clear validation also squared out in the very first maybe portion of phase 1 that you can use later on for the expansion phase. Nowadays, as you know, we really don't even have specific phases of clinical development. Starts with phase 1, and if the drug is highly effective, it's going to go fast down the development path. So you have to be ready, you have to understand the assay, the target, and that will determine the level of investment and development, even in phase 1 in oncology.
Thank you so much, Suzanne. I know we're a little over time, so I'm going to let everybody go. Just to reiterate, we will be sending out the slides as well as the recording, and then at that point you can reach out if you'd like a consultation or have any additional questions. Thank you so much for joining us today, and have a great afternoon.